So good afternoon. This is Nelly Poyak from Dana Farber. Thank you everyone for joining us at this the FHCC seminar series, Connect Science. And as a reminder, we posting the presentations on the website that's indicated on this first slide. And just in case you run into issues with viewing the slides, if you're using Firefox, try to use a different browser. Some people had issues with this. And again, as a reminder also, we will have a Q&A session at the end. Please post your questions in a Q&A function on Zoom. And we try to answer as many questions as we can live, and then or the ones that we don't have time to answer during the presentation, the speaker will have access and he can get back to you and answer the questions. So with that, it's my pleasure to introduce our colleague from Dana-Farber, David Pellman. David is a Margaret Dyson Professor of Pediatric Oncology at Dana-Farber Cancer Institute, and he is a Professor of Cell Biology at Harvard Medical School. He's also an investigator of Harvard Hughes Medical Institute and has expertise in aneuploidy and in cancer. And today, David will discuss his work on the origin of complex cancer genomes. Thank you, David.
Great. Okay, thanks so much, Nelly and David, for organizing this wonderful seminar series. And thanks for the invitation. So as Nelly mentioned, I'm going to talk about our work on the mechanisms of cancer genomes. I'm going to try to take a broader scope of our work, and also sprinkle in some unpublished data. And then I'm going to also end with a brief summary of a paper that by happenstance is just published out today in the current issue of Science. So my disclosures, I have some company connections, but none of them are relevant or influence the talk I'm going to give today. So here's the question that we're trying to understand, which is how do you go from a completely normal genome, human genome with 46 chromosomes, to a cancer genome that could have hundreds, thousands, or even in this extreme case, tens of thousands of chromosome rearrangements. This is an issue that we thought that we sort of knew the answer to. We thought it happened in the way that Darwin had taught us, meaning that small-scale genetic changes would happen one generation after the next, and that these genetic changes would slowly and gradually accumulate, taking you from the normal genome to the altered complex genome. However, if you just sit back and look at this cancer genome and think about all of the rearrangements going on, you could start to wonder how long would it really take if this happens slowly and steadily, one step at a time. As an alternative, there's been a new idea that's emerged in the field that my group has been very involved in, and many others as well, which is that instead of going slowly and steadily, genome evolution of cancer may occur in sudden bursts, where large mutational events that generate showers of many chromosome alterations happen all at once and episodically. And we find this idea appealing because it can parsimoniously explain with a small number of catastrophic events how you get such a highly complex genome. Of course, those ideas, I should say, are not mutually exclusive and could be happening together at the same time. One large-scale theme that's emerged from our work is that complex alterations in the genome are stemming in large part from abnormalities in the architecture and the integrity of the cancer nucleus. What I've illustrated here is an extreme case of a cancer cell line that has extensive so-called nuclear atypia. So abnormal shapes of the nucleus, little blebs of the nuclei, little structures that we'll talk about and then other abnormal structures connecting divided cells called chromosome bridges. Abnormalities in the shape and architecture of the nucleus have been noted in cancer since the late 1800s. And since the 1920s, pathologists have used the degree of nuclear atypia as one component for assigning tumor grade and hence predicting patient prognosis. So the biological and medical significance of abnormal nuclei in cancer has been known for a long time, but precisely what goes on in these abnormal nuclear structures, how they're different from regular nuclei and how they might influence the biology of tumor cells has been much less studied. And that's something that we're trying to address. So there's at least 3 major processes that are known to drive rapid genome evolution, and these are 3 that my lab has spent a lot of time working on. I'll briefly mention early work from my lab on the duplication of whole genome and its contribution in cancer. And then we'll talk about a catastrophic mutational process called chromothripsis, a neologism for chromosome shredding. And then at the end, we'll talk about a process called chromosome breakage fusion bridge cycles, first described by Barbara McClintock, and we'll discuss its relationship between chromothripsis and breakage fusion bridge cycles. There is a 4th major process that was discovered by Levi Garraway's group called chromoplexy, and that I won't have anything to say about. Okay. It's hard to talk about chromosomes in cancer without starting with Theodore Boveri and his famous work and hypotheses. So Boveri was an originator of the chromosome theory of inheritance and also discovered an important organelle in the cell called the centrosome that forms the poles of the mitotic spindle. And as illustrated here, Boveri created a variety of manipulations that altered the number of chromosomes in cells, giving you an abnormal mitosis, as is illustrated here, and then giving you, which then results in abnormal chromosomes segregation, and daughter cells with different compositions of chromosomes. And in a series of papers around 1905, Boveri and his wife showed that chromosome missegregation gave different phenotypes, which was sort of the foundation for his chromosome theory of inheritance. In addition to that, it was known that having too many centrosomes in cells would give you these multipolar mitoses, and others had observed these kind of abnormal mitotic figures in cancer cells. And so in this famous monograph, Boveri proposed that extracentrosomes, whole chromosome missegregation resulting in aneuploidy might be the origin of human cancer. And one of the major ways that was easy for Boveri to generate cells with extracentrosomes was to inhibit the last stage in cell division called cytokinesis, which doubled both the numbers of chromosomes and also the numbers of centrosomes in the cell. And one of the many interesting proposals in this monograph was that cytokinesis failure and whole genome duplication might be a driver of human tumor genesis. So that idea sort of percolated through the field over many decades and was alternatively forgotten and resurfaced. Many interesting, intriguing early studies citing correlations between genome doubling and cancer, but there had been no simple and direct test of the idea that cytokinesis failure could drive tumor. And a paper from my lab, Fujiwara, led a study where we isolated primary p53 minus mammary epithelial cells from a mouse, induced the failure of cytokinesis with drug treatments, separated the resulting tetraploid cells from unaffected diploid cells and transplanted them into animals to test the hypothesis that genome doubling could drive tumor development. And this experiment had sort of good news and bad news. The good news was that the animals that got the genome doubled cells did indeed develop tumors rapidly with high penetrance. And the chromosomes were wildly abnormal. And consistent with our hypothesis that whole chromosome aneuploidy might be contributing to tumor development a la Boveri, we did see lots of whole chromosome aneuploidy in these tumors. However, there was a problem, which was that we didn't only see that, and we saw lots of evidence of DNA breaks and chromosome rearrangements, like this non-reciprocal translocation illustrated here, and other evidence of things like double minute chromosomes. So the simplest hypothesis that whole genome duplication would only give you pure aneuploidy wasn't applicable in this system, but this actually raised another interesting point, which is that the cytokinesis failure should give you pretty much pure defects in mitosis because of the extra centrosomes. So that raised, but how did you get all this chromosome rearrangement? This raised an interesting possibility that errors in the missegregation of intact chromosomes might somehow lead to chromosome rearrangement. So hold that thought. At the time that we did that experiment, our experiment established that cytokinesis failure could, in certain experimental circumstances, give you tumor development, but it didn't say in real human tumors how frequently that is. But that's now been addressed by many other labs through genomic analysis, and we now know that whole genome duplication is a major feature of human cancer, happening in something on the order of 40% of all human tumors at some point during their evolution. That doesn't mean that in every case it's causal, but it does say that whole genome duplication is a major feature underlying the architecture of many human cancer genomes. Okay, so let's return to something that an abnormal mitosis, shown here with this cell with 4 centrosomes, could somehow lead to DNA damage. And the idea was appealing, but we needed a hypothesis of how that might work. And because of work that we were doing to study the consequences of having too many centrosomes in cells, and that was in this paper by Neil Ghanem from my lab, we developed an idea, which in this paper, which really addressed how chromosome attachments go wrong in cells that have failed cytokinesis and have too many centrosomes. What we found is that, actually different from what Boveri hypothesized, and rather than the cells dividing in 3 or 4, sort of going off like a bomb, instead what happens is that cells can actually bundle up extra centrosomes, and that allows them to divide in 2, but there's a price to pay, which is that the abnormal geometry of the spindle sets up abnormal attachments of microtubules here in green to the red kinetic cores, which can result in chromosomes essentially being caught in a tug of war and lagging behind in anaphase. This is a cell that's separating 2 duplicated sets of chromosomes here with a lagging chromosome. And if the chromosomes lag enough, when the nuclear envelope reassembles in the next interphase, these chromosomes can get left out sort of in the cold in their own nuclear structure called the micronucleus, illustrated here. So that got us wondering about the actual functional integrity of these nuclear structures relative to a normal nuclear structure, and sort of motivated a hypothesis that was also inspired by a rather complex early literature, that maybe these little micronuclei could be like mutator organelles that damage intact chromosomes, giving a connection between mitotic errors and DNA damage. So Karen Cresta from my lab in 2012 did a very simple experiment to test this idea, where she created these kind of missegregation events de novo, and then watched the formation of a micronucleus without any pre-existing DNA damage. That was a big issue in some of the early studies. And then she just waited and watched to see what happened. And this cartoon here shows what she found, which was that in a very high fraction of cells, around 50% of the cells she was looking at, she would then see over time the appearance of very massive accumulation of DNA damage that, for example, phosphorylation of gamma H2AX. So this was satisfying because it suggested indeed that missegregating chromosomes into these abnormal nucleus structures could give you very extensive DNA damage. And the mechanism underlying this damage is a very interesting and important issue now in the field. I'm going to talk about some unpublished results on that in a minute. But a very important insight came from another group. Emily Hatch and Martin Hetzer's group showed that the nuclear envelope surrounding these micronuclei is fragile and prone to spontaneous rupture, spilling the nuclear contents into the cytoplasm. Which then somehow leads to extensive DNA damage by a mechanism that I'll address in a bit. I won't have time to talk about this, but this is a fascinating phenomenon that also exposes DNA into the cytoplasm and can activate innate immune signaling by the C-gas sting pathway, which is now of great interest in cancer therapeutics. Okay, while we were working on that paper, Peter Campbell from the Sanger Institute published a paper describing a phenomenon that he identified through comprehensive whole genome sequencing of cancers that he called chromothripsis. And chromothripsis is essentially massive rearrangement of one or only a few chromosomes that looks in essence as if somebody took a chromosome illustrated here and with an order from A to H, blew up this chromosome into fragments, and then randomly took those fragments and stitched them back together again in a way that creates a novel so-called derivative chromosome. This, I think, was one of the more surprising findings from comprehensive cancer genome sequencing, and it was a big puzzle of how you would get massive mutagenesis on only one or a few chromosomes, and the mechanism obviously wasn't clear. Again, this just illustrates what this looks like, the fragmentation of reassembly of the chromosomes like shuffling a deck of cards. We now know that chromothripsis is also, like whole genome duplication, quite common in cancer, maybe 20–30% on average in epithelial tumors, and with some cancers like pancreatic cancer with frequencies as high as 65%. Oftentimes, these pancreatic tumors would have more than one chromothripsis events, explaining something like 50% of the rearrangements in these tumors. So a good example of a single catastrophic event that generates massive mutagenesis. Although this phenomenon was first discovered in cancer and is particularly prominent in cancer, it's in no way specific to cancer or indeed humans. Chromothripsis can happen in the germline. It can cause rare human congenital disease. It can actually be passed through the germline, so in principle, it could be contributing to organismal evolution, and it's been shown to occur in plants in very interesting circumstances by my colleague Luca Comai that I won't have time to discuss. Okay, so we had showed that the formation of micronuclei could give you very specific isolated DNA damage of the chromosome that was in the micronucleus, and it was therefore a rather simple and straightforward hypothesis to wonder whether the DNA damage that we observed in micronuclei could explain chromothripsis. So that's an appealing hypothesis, but how do you test that? Obviously, the micronucleation is a cell biological phenomenon, and chromothripsis is a phenomenon you can only see by sequencing genomes. And so that led us to the obvious need to set up a method where we could manipulate cells in culture during long-term imaging, say over 2 or 3 days, create, in this example, micronuclei, watch the envelope break as a surrogate for the formation of DNA damage, watch the damaged chromosome be reincorporated into daughter cell nuclei, and then come in. The way that we do this now is with a microcapillary robotic arm, and pick up each of the daughter cells individually and prepare them for single cell whole genome sequencing. This is published, and I'm just going to show you a movie that summarizes this work. Okay, so we can make micronuclei from mitotic errors. For example, if you have too many centrosomes causing misattachment of chromosomes, so-called merotelic attachments for the cognoscenti, causing chromosomes to lag and forming micronuclei. It's important to note that any single unrepaired DNA double-stranded break can cut off the end of a chromosome, which then won't be able to attach to the spindle, and the same thing happens. So many common cell division errors give you micronuclei. We and others have shown that there's very poor DNA replication in these structures. The nuclear envelope is fragile and breaks. Somehow, mysteriously, exposure of the chromosome to the cytoplasm leads to DNA damage and chromosome fragmentation. And if the fragments of the underreplicated chromosome are dispersed, and then we pick up the cells and sequence them, and here we're going to look only at the haplotype of the affected underreplicated chromosome. And what you can see is that this experiment essentially recreated all of the known features of sequence features of chromothripsis. So this is an affected chromosome. This is the affected copy number of the affected haplotype. You can see that fragments of the chromosome are either present, absent, present, absent, present, absent, meaning that you get the very characteristic oscillating DNA copy number pattern that was the original puzzling thing that brought chromothripsis to attention. And these links are the rearrangements. And a very important part of our analysis that I don't really have time to go into in great detail, but I'll give a shout out to, is the fact that we compare the sister cells means that we can see that if a fragment is present in one cell, it's generally missing in the other, present in one cell, the other. So the mirror image copy number pattern here is a really nice biological validation that the sequence changes that we see are true. And a lot of this was built up by my former trainee and now an independent investigator and collaborator, Chengzong Zhang of the Data Sciences Department at Dana Farber. Okay, so now in terms of the mechanism of chromothripsis, we can simplify a bit and break it down into a series of steps. I've told you that one way to get chromothripsis is to create these abnormal nuclear structures called micronuclei. The next question that comes up is, why is the nuclear envelope fragile and why does it spontaneously rupture? And then the initial rupture of the chromosome in interphase appears to lead to extensive DNA damage, and the mechanism of that has also been unclear. And for reasons that I'll discuss a little bit later, we think there are actually two big waves of DNA damage. One that happens when the envelope ruptures, and second, when the chromosome from the micronucleus is exposed to the cytoplasm of the cell in mitosis, when that cell advances into mitosis. And I'll discuss that and what we know currently about that mechanism. And these things, then you have to stitch up the fragmented chromosomes, and presumably that can give you chromothripsis. Okay, I don't have time to discuss the nuclear envelope in very great detail. We published a paper on that proposes at least the beginnings of an understanding of a mechanism for the defect in the nuclear envelope. But I just want to show you one piece of data. What we found in this study by Shiwei Liu and Mijung Kwan from the lab is that when the nuclear envelope is assembling on chromosomes that are lagging and ultimately might be in micronuclei, what we found is that certain nuclear envelope proteins assemble normally on the lagging chromosome, like this protein here called emerin. However, there is a striking defect in the assembly of other key nuclear envelope proteins, such as the nuclear pore complexes that mediate the transport of proteins in and out of the nucleus. And the point that I want to make here is that this defect in nuclear pore assembly, which leads to defects in nuclear transport and DNA replication and transcription, we think that this envelope defect is actually sort of the original sin of this whole process here, which sets up the entire downstream cascade of events that we'll be discussing in a minute. Okay, now I'm just going to give you a brief note on some work in progress about why the interface cytoplasm is resulting in DNA damage to the chromosome from the micronucleus. Here, we tested a bunch of specific hypotheses, and with my colleague Wade Harper in the Department of Cell Biology at HMS, we also did a proteomic analysis of the contents of micronuclei. And in part based on this proteomic analysis, and in part based on hypotheses, and in part based on a little bit of luck and some also very hard work by Shangming Tang in the lab, we've formulated a hypothesis that the DNA damage in micronuclei may result, at least to some significant degree, from aberrant DNA base excision repair. Just to bring everybody up to speed, this cartoons an outline of DNA, a crude outline of DNA base excision repair. The base excision repair pathway recognizes abnormal bases with small adducts, and there are 11 glycosylase enzymes in the human genome that scan DNA, recognize abnormal bases, flip them out from the DNA double helix, and alone or in concert with other enzymes, then cleave the N-glycosidic bond to the DNA backbone, leaving an A-basic site. This A-basic site is then recognized by the AP endonuclease, APE1, which generates then a single-stranded DNA nick. Of course, normally what happens is that there are two downstream pathways that lead to high-fidelity repair of these lesions. But the important point for this talk is that if this repair doesn't go properly, the DNA nick can be converted into a DNA double-stranded break, as we think happens in micronuclei, and would be obviously of the chromosome fragmentation leading to chromothripsis. And that can happen in one of two ways. First is if you have closely spaced nicks on opposite strands, that can be converted into double-stranded breaks. This, for example, is what occurs in vitro recombination. Additionally, if you just undergo, if you do DNA replication through a nick, you can see here would be the leading strand of a replication fork. Here, when it runs into the nick, it generates a one-ended DNA double-stranded break. So DNA nicks can lead to DNA double-stranded breaks. We've now done a variety of experiments to show that APE1 is for the majority of DNA damage in ruptured micronuclei. And I'm just going to show you one experiment that I think is the most straightforward and convincing, which is that we appended with CRISPR the APE1 gene with tandem degron tags for AID and SMASH, and edited both copies of APE1 in the genome. That allowed us to arrest cells in G2, get rid of APE1, which you can see here on the western, and release these cells in the presence of drugs that would interfere with chromosome segregation, generate micronuclei, and then measure the fluorescence intensity of DNA damage. What you can see here is that there's a very significant reduction in DNA damage in these ruptured micronuclei. So of course that made us wonder whether APE1 actually binds to these ruptured micronuclei, and the answer is that the wild type protein doesn't. That actually was not completely unexpected because enzymes act catalytically on their substrates and are generally built to turn over rapidly and dissociate from their substrates. But APE1 has been studied very extensively, and you can and there are mutants in APE1 that specifically affect the off rate of the enzyme for its substrate without affecting the enzymatic activity. And you can see that this mutant with the slow off rate accumulates in a very high percent, like 70% of the ruptured micronuclei. So the enzyme can accumulate on and the damage is dependent on APE1. And this has opened up a whole host of interesting questions for us. So presumably, since it's the base excision repair process, this suggests that there may be abnormal bases on micronuclear chromosomes, which is something that we're pursuing. And knowing what those are, we'd like to understand the cause of those abnormalities, identify the relevant glycosylases. And then finally, this gives us a handle. DNA damage has been reported to occur in primary main nuclei in a variety of circumstances, such as when tumor cells are squeezing through tight spaces at the beginning of metastasis, or hypothesized to be so. And we'd really like to understand whether the DNA damage in other circumstances where the nuclear envelope is rupturing is similar or different what's going on in micronuclei. Okay, so that's one wave of DNA damage. I told you that there's a second wave of DNA damage when that cell with ruptured micronucleus advances into mitosis. And we investigated this largely because of a really interesting older literature that's illustrated by this famous experiment by Johnson and Rao here. What this was Johnson and Rao were studying the cell cycle and which phase of the cell cycle would be dominant over others, and learning something about the organization of the cell cycle based on that type of experiment. And in the experiment shown here, they took a mitotic cell and fused it to an S-phase cell here and described a phenomenon that they called chromosome pulverization, where they hypothesized that the mitotic cytoplasm might be fragmenting the S-phase chromosomes and causing this pulverization phenomenon. And the reason that we found this interesting is that, based again on old literature and our own experiments, the micronuclei replicate DNA very poorly, but even in G2, they keep trying. So this is a G2 cell that's been pulse labeled with BRDU, and you can see the micronucleus is still trying to replicate its DNA. And then when the cell advances into mitosis, the micronuclear chromosome essentially undergoes the same kind of chromosome pulverization phenomenon. And when Peter Campbell's paper first came out, Matthew Meyerson and I wrote a mini review accompanying that paper where we speculated that this kind of chromosome pulverization phenomena might have something to do with the mechanism leading to chromothripsis. And indeed, we think that we now have evidence that that's the case. So this is an experiment where we looked specifically at DNA, asked the question whether DNA damage happens on these micronuclear chromosomes in mitosis. What was done here is that in synchronized cells, we arrested cells here in the G2 phase, and we looked only at cells that had intact micronuclei. I'm not showing you the data supporting that. You'll have to take my word for it. And then we pulse those cells with EDU. You can see that there's very little DNA replication as expected and little DNA damage by gamma H2AX. But if we now take this population of cells and release them into mitosis, now specifically in the presence of EDU that's the cells were only exposed to in mitosis, so any DNA replication that you see is only happening in mitosis, we saw something really rather remarkable, which is that when the chromosome from the micronucleus in the cell goes into mitosis, what you get is a huge burst of DNA replication specifically happening on this chromosome. And that is associated with massive amounts of DNA damage. I'm not showing it to you here, but there's also massive amounts of single-stranded DNA. Okay, so there's a second wave of DNA damage that's happening here, and it's very extensive. If we sequence the cells in G2, consistent with there being little DNA damage, only about 10% of these cells show any evidence of chromothripsis. But if we now let these cells pass through mitosis and go into the next G1 phase and then grab the daughter cells, we found in almost every case that the daughter cells exhibited chromothripsis. So transit through mitosis is something that promotes chromothripsis on these micronuclear chromosomes. So this is a fascinating phenomenon addressing what's the nature of this replication, what's causing the DNA damage. These are all very interesting questions. However, addressing the mechanism of this damage is somewhat challenging because in tissue culture cells, there's a very short time interval of mitosis, and you really have to synchronize cells and do very precise experiments. And so to circumvent that, I joined forces with my friend and colleague over in the biochemistry department at Harvard Medical School, Johannes Walter. And what Johannes and I did was to use his elegant cell-free in vitro system based on Xenopus egg extracts, where he can get DNA replication to go in completely cell-free and nucleus-free extracts. And then that can be done with plasmid substrates, and we can take those extracts and drive them into a mitotic-like state simply by adding mitotic cyclins and cyclin-dependent kinase 1. And again, this paper was published last year, and I'm not going to show you any primary data, but I'm just going to show you the model. The results of this work shows that if you have a stalled DNA replication fork, which is essentially what we expect happens in micronuclei. They have poor nuclear import and abnormal nucleoplasm and few replication proteins. So the replications start but run out of gas and lead to arrest at either a barrier or just spontaneously stop. And then if the cells enter mitosis with that under-replicated DNA, what we found is that a little studied E3 ubiquitin ligase called TRAPE adds ubiquitin molecules to the replicative helicase called CMG, and perhaps other replication proteins at the fork. And what that does is create a signal that leads to an ATPase called the P97 ATPase that then pulls off the ubiquitinated helicase from the replication fork, which basically unravels the entire replicosome and triggers the unloading of the helicase and the rest of the replisome, leading up to a little piece of naked DNA that now becomes a substrate for an unknown nuclease, which is then cut, and now we have broken DNA ends that can be ligated together by alternative end joining or other possible mechanisms. So what we think happens is that if there's just a few under-replicated segments of DNA, as you might see in famous sites in the genome called common fragile sites, this type of controlled DNA replication fork breakage might give you a solution to get some kind of effective chromosome segregation with the sacrifice of making a few breaks. But if you had a circumstance like in a micronucleus with many stalled replication forks, you'd wind up with something like this, where you'd have many forks, many breakage sites, and then those breakage sites could then generate fragments that might be rearranged and lead to chromothripsis. Okay, so just to summarize, there's a series of events that are happening in micronuclei that are leading to chromosome fragmentation and contributing to chromothripsis. Now, in the last part of the talk, I want to just turn to another phenomenon and address the question of whether there's really one specific mechanism for chromothripsis that's common and general, or are there really separate, or are there separate ways of generating chromothripsis from different, perhaps from different nuclear structures? And I'm going to turn to these structures here, which are chromosome bridges, which can form either by failing to decatenate chromosomes, or can be caused by chromosome fusions, for example, which would happen if you have critical telomere shortening or dysfunctional telomeres, or from incompletely replicated chromosome ends. In many, like micronuclei, chromosome bridges are a final common product of many errors in cell division. Okay. The interest in chromosome bridges in cancer goes way back to a famous series of papers by Barbara McClintock, where she proposed that the so-called breakage fusion bridge cycles involving chromosome bridges would be a mechanism that could lead to gene amplification. This is my doctored version of the summary scheme from a paper by McClintock in 1941. And she was working in corn, maize, and generated broken chromosome ends by radiation. And here's her hypothesis, that if you have a broken chromosome end in a cell that's in G1, and then that cell undergoes DNA replication, the two replicated sister chromatids will now have broken ends right adjacent to each other before the cell goes into mitosis, and the broken ends will now fuse, as illustrated here. When the cell goes into mitosis, the centrosomes, the centromeres, illustrated here by the open circles, will get dragged between the dividing daughter cells like this. And then once you have a fusion here, you have, of course, no memory of the original site where the fusion occurred. So any random breakage now is going to give you asymmetry. So what you can see is that one daughter cell here is going to get two copies of the A segment in an inverted orientation, and the other daughter cell is going to get a reciprocal loss of that segment that was gained here in one daughter and it's lost here. And I hope you can appreciate that if you iterate cycles of these breakage fusion bridges, what you'll see, what you can get is an exponential amplification of the terminal segment. You'll go from 2 to 4 to 8, 16, etc. So it's been widely assumed that chromosome breakage fusion bridge cycles, particularly due to critical telomere shortening, are a major origin for oncogene amplification in human cancer. And if you look in real cancer genomes, you can certainly see evidence for these kind of breakage fusion bridge cycle events. But the kind of puzzling point is that you don't very frequently see the simple version of the BFB model that was first proposed by McClintock. Instead, you see much more chaotic structures that might be intermingled with the BFB pattern. What hasn't been clear is whether the added complexity is something that happens later that's obscuring the original simple BFB pattern, or whether the BFB model itself, process itself, is intrinsically more complex than originally envisioned. So we got interested in understanding a potential relationship between chromothripsis and breakage fusion bridge cycle based on this work by others. Peter Campbell and Christine Harrison showed that in some specific circumstances that chromothripsis and breakage fusion bridge cycles could co-occur. And indeed, experimentally, if you induce chromosome breakage fusion bridge cycles in vitro, you can also get an outcome in populations of chromothripsis. So these experiments raise the interesting possibility that maybe chromothripsis is somehow mechanistically entangled with the chromosome breakage fusion bridge cycles. And in the paper that's just out today, we've addressed this using a combination of cell biology and then also our single cell sequencing experiments to see the immediate outcome of chromosome bridge breakage, and then also couple that with long-term population growth experiments where we can see the consequences of chromosome bridge formation in a single population over many generations. And here I'm just going to summarize a rather complex story briefly and then just hit a couple of points that are relevant here. So the first point is that I told you that there's kind of a cascade of events leading from a micronucleus to the generation of chromothripsis. And many of those events are also conserved and baked into the chromosome breakage fusion bridge process. So the chromosome bridges, although they look different from micronuclei, they actually have the same defects as micronuclei in their nuclear envelopes, the same defect in nuclear pores, the same defect, same apparent defect in DNA replication, and the same level, low level of chromothripsis associated with the initial formation and breakage of the chromosome bridges. In addition to that, the little stubs of the broken chromosome bridge essentially behave identically to micronuclei when you go into the next cell cycle, and also wake up and give a burst of DNA replication that's associated with extensive DNA damage. But there's two things that are a little bit different. The first is which I'll turn to in a second, is how are the bridges broken? And then the second point that's also relevant is that these broken chromosomes appear to have problems with their centromeres, so that the chromosomes don't attack properly. In the second generation, when you're making granddaughter cells here, actually generate micronuclei at very high frequency. So the chromosome bridge triggers a whole cascade of events that leads to a series of DNA damaging events, including the downstream formation of micronuclei that could give you essentially very rapid onset of high genome complexity. Okay, so now I'm just going to mention some things that are different, and then I'll summarize. One thing that's different is that the chromosome bridges are actually physically broken, and I want to just briefly mention our model for that. Well, you can see here, these are two cells. You can't see the other cell completely up here. And you've got, the green is showing you a label for the DNA, and here's the chromosome bridge. What you're going to see here is we're just going to watch the chromosome bridge break. And the point that I want you to take away from this is that it appears as if cell protrusions are reaching out and grabbing the bridge and then pulling it tight, and then the bridge is going to break under the mechanical force of the apparent pulling forces. You can see that here. So there's a red stain is the cytoplasm, which you can see is sort of crawling out along the bridge, pulling the bridge taut here, and then the bridge breaks, and you can see the stub of the chromosome bridge. I'll play that one more time. So this data, this is of course purely correlational, but we have functional tests of the requirement for actin and myosin that's in the published paper. And what we think here is happening here is something very interesting, which is that the contraction forces of actin and myosin, in other words, like the contraction of a muscle, is actually creating mutagenesis by physically contributing to the breakage of the DNA that's in the chromosome bridge. And one way that we did that is to let these bridges form and then put in inhibitors of either actin or myosin, and we can essentially near completely block the breakage of the bridges. So actin and myosin is required. It may or may not be sufficient, but it's clearly necessary to break chromosome bridges. And if we pick up those cells with the spontaneously broken chromosome bridges, we can see a variety of events. Sometimes we see the simple pattern of reciprocal gain and loss, which is predicted from the McClintock model. But other times we also see more evidence of a kind of local fragmentation. Little bits of fragments that are retained. This is in the segment of the chromosome bridge here. And you can see that these have been rearranged. So you can see little low-scale evidence of a chromothripsis-like pattern happening here. And we think that it's possible that the mechanical force itself may not only give you simple breaks, but some local fragmentation. And that's based on an experiment like this, where here is a cell with a chromosome bridge, and we're coming in now with a microcapillary. And in the movie, you're going to see the capillary come in and break the chromosome bridge, and then we're going to come in and pick up the daughter cells and sequence them. Okay, so here's the capillary. It's stretching the bridge. Now the bridge breaks, and then we're going to grab these two cells and then sequence them. What you can see is that the mechanical breakage of these structures essentially recapitulates all of the features that we saw in the spontaneous breakage. So there are two daughter cells here. You can see the reciprocal gain and loss in a simple rearrangement here. Here's an example of simple fragmentation. Here's another example of more extensive fragmentation. So this initial step is a little bit different from micronuclei, but the rest of the cascade is rather similar, and we'd like to propose that by and large there are unifying themes in how abnormalities of the nucleus generate chromosome instability. Okay. So finally, if this whole cascade and the subsequent production of micronuclei and further cycles of bridging is really all true, what you would predict is that if you use CRISPR, as we did here, to create a defined chromosome 4 bridge, watch that bridge spontaneously break, and then isolated the individual cells, and instead of sequencing them, we sent them off and let them grow into populations. What you might then recapitulate is a cascading effect of ongoing complexity and ongoing evolution, leading to additional changes and much subclonal heterogeneity, as you would expect to see, as we do indeed see in true cancer genomes. And because time is short, I'm just going to very briefly overview this data. So here what we've done is to make these populations, and then we clone them again into subclones. Here's the sequence of the bulk population. And there are two, there's the one homolog is in blue and the other homolog, the copy number is in red. The breakage from the CRISPR cut was here. And what you can see here is that the red homolog here is present in a quite variable copy number. So it should be present either in 1 copy, 0 copies, or 2 copies, but the intermediate copies are essentially telling you the whole story of what's going on here, is that you have quite extensive subclonal heterogeneity with some cells in the population with 0, 1, or 2 giving you all these intermediate states. And I'm just going to, we confirm the idea that there's extensive subclonal heterogeneity by single cell sequencing and also by bulk sequencing of subclones in these populations. And I'll refer you to the paper for the details. Okay, so just to summarize here, we think work from our group and many others, now the paths that are leading from a normal genome to a highly complex cancer genome are starting to come into focus. And we're starting to have a much better understanding of some of the catastrophic mutational processes, such as chromothripsis and breakage fusion bridge cycles that are leading to cancer genome complexity. I hope I've also convinced you that by understanding the pathological processes, such as the defect in nuclear envelope assembly, that we can shed some light on fundamental questions in normal cell biology, such as how the normal mechanism of nuclear envelope assembly actually works and why it would go wrong for a lagging chromosome. And then finally, in the very last section, we're trying to understand how these different catastrophic mutational processes are interrelated. In other words, like how the BFBs and chromothripsis are interrelated. And this analysis is starting to give us some sense of the rules of the road that are governing the evolution of complex cancer genomes. Okay, I'll stop there. I reviewed too much work to thank everybody from my group over quite a long time that's contributed to this work. But I'm going to give a shout out to the people who did the recent unpublished or recently published work. The work on the base excision repair is spearheaded by a talented postdoc, Shangming Tang. Neil Umbright, who's a crackerjack cell biologist and biophysicist, drove the work on chromosome bridges with help from many others in my laboratory. Particularly, I'll give a shout out again to Chengzong Zhang, my collaborator on the genomic analysis. And we've had wonderful collaborations with many labs, including Dipanjan Chowdhury, Matthew Meyerson, Alexei Kojukov, and this is wrong. It should be JW or Johannes Walter here. So I'll stop, take questions. These are funding and affiliations. Thank you.
Okay, thank you, David, for the outstanding talk and beautiful movies that really make you see what happens. So we kind of short of time, but we have a number of questions, and several of them related to the bridges. Like, is there any particular DNA sequence that, or like epigenetic pattern that would predict like where would the bridge and breakage would be? And do these only occur in tumor cells? And are there any particular tumor types that are more likely to have this than others?
McClintock's original work was done in corn. So bridges can happen in any cell type. The bridges can happen by fusion in any chromosome, anywhere, giving you a fusion or entanglement close to the ends of that chromosome. So there's no specific chromosome that is targeted into the bridge. The bridges are super common. If you just do a DNA stain in Joe average cancer cell line that anybody out there is doing their experiments in, you'll see single-digit percentages of cells, typically with both micronuclei and chromosome bridges.
Okay. And do you have any particular, like, is there any particular force on these bridges? And what determines whether the cell after such a mitotic catastrophe survives or not? And what is their fitness advantage if they do survive?
Okay, this is a very important point. We believe that what I try to convince you of is that there are actomyosin contractile forces that are pulling on the bridge. How that actually works is not clear, but we have some evidence that this is force from the cytoplasm that's transmitted from focal adhesions through actinomyosin to the nuclear envelope, and then attachments at the nuclear envelope would transmit that force to DNA. The other question that you asked is a very important one, which is that how frequently do these events lead to the establishment of an abnormal chromosome in the population, and what fraction of them lead to a high frequency of cell death? I mean, in general, these are huge mutations, huge mutational processes, and we assume that almost all of these events would compromise cell fitness and maybe even be lethal. And that it would only be the rare events that would lead to the amplification of an oncogene or the inactivation of tumor suppressors that would give an advantage that would lead to cell growth. The data that chromothripsis, for example, can lead to oncogene amplification and inactivation of tumor suppressors is now extremely convincing. And I would, for example, refer people to the Peacock papers and the summary paper from Campbell's group. So there's extensive evidence that the chromosome fragmentation can simultaneously take out multiple tumor suppressors. Many of the oncogene translocations that are, for example, in Ewing sarcoma and in many epithelial tumors are driven also by chromothripsis. And so-called double minute chromosomes, which can, like breakage fusion bridge cycles, can give you oncogene amplification, or also there's growing data that these structures can derive from chromothripsis and the circularization of chromosome fragments.
Okay, thank you. We have many other questions, and David will have a chance to look over these and get back to you because we ran out of time now to answer more. So thank you again, David, for the outstanding talk. And thank you, everybody, for joining us for this seminar, and have a great weekend. We will have a seminar next Monday, despite that that's a Massachusetts holiday, but we continue the series. And so hopefully we'll see you, many of you, Monday at the usual time. Thank you so much. Bye.